Good afternoon, everyone. If you could have it nice and quiet, please. So on behalf of the Cumming School of Medicine, it's my pleasure to welcome you to the 4th annual Friesen Lecture. As you know, we're celebrating our 50th anniversary as a medical school this year, and I think from the get-go, the Faculty of Medicine, which is now the Cumming School of Medicine, has always prided itself in research. We've made some great accomplishments in that area, and I believe that we've had a tangible benefit on the well-being and health of Albertans because of the activities that happen in our school. So today we are very pleased to welcome Dr. Janet Rossant as the 2016 winner of the Henry Friesen Prize. Now, this prize was established in 2005, and it is really there to recognize excellence in science. And Henry Friesen was a Canadian endocrinologist who discovered prolactin, and he was also the president of the Medical Research Council just before it became Canadian Institutes of Health Research. So he's had a major impact on the research landscape, and so it's a very worthy prize to be named after him. So Dr. Rossant spent her day today meeting with a number of stakeholders, including members of the Charbonneau Cancer Research Institute, the Alberta Children's Hospital Research Institute, and the Leaders in Medicine program. And it's been really a privilege to hear her thoughts on the research landscape and the challenges that we face and maybe some solutions. So it was a real pleasure. I would like to also take the moment to acknowledge Dr. Aubrey Angel, who is the president of the Friends of CIHR and also a senior fellow at the Massey College, and he's instrumental in making this lectureship circuit happen. And so thank you very much for being here and coming along. And now I would like to invite Dr. John Reynolds, the Associate Vice President for Research of the University of Calgary, to say a few words.
So thanks very much, Gerald. It's a great pleasure to be here this afternoon, and I'm delighted to have this opportunity to welcome Dr. Rossant as well to the University of Calgary. You're such a distinguished scientist. I think you have probably one of the best jobs at the moment as president of the Gairdner Foundation, and who amongst us would not take the opportunity to spend a lot of our time speaking with some of the strongest, best scientists that are having an enormous impact on healthcare in the country and in the world, in fact. So very envious of that opportunity. And I was at the last Gairdner Foundation award night that was, I think, your inaugural evening, and a wonderful event filled with CRISPR Cas9, incredible stories of success and importance in research. So thank you for taking on that important role as well. Just as a little opportunity to indoctrinate you perhaps in what's going on at the University of Calgary, but a number of years ago we set our eyes on a very bold and ambitious future for the university. It was encapsulated in the Eyes High initiative. Part of the intention is to become one of the top universities in research universities in Canada, and to achieve this, we designed a research plan that identifies areas of societal needs where we believe we can make a big impact and help society. As Gerald mentioned, this is the 50th anniversary of the Cumming School of Medicine, and the University of Calgary sees medical research as being a priority of the university, and it's reflected in our commitment of building leadership in the various areas of medical research, and we've identified a number of them, brain and mental health being one, inflammation and chronic disease is another, cardiovascular health, bone and joint health, child health, and cancer research, of course, and finally, last but not least, public health. So these are all important areas to the University of Calgary. We've identified them as priorities and looked for ways to invest to ensure that the opportunities that are available for research here will optimize the outcomes, both for the research and for the health of Canadians. So we focused on these selected areas of strength, and we're building a critical mass in research and creating a dynamic research environment to ensure that our work makes a true difference. Our scholars are addressing some of the biggest challenges of our time, and we are proud to have some of brightest minds in the world in research at the University of Calgary. This year is the start of what we call Energizing Eyes High, and this is the next round after the first 5 years of that program, which I think has shown significant success in creating what we envisage as one university, and many of the opportunities are aimed at bringing the best and brightest researchers from different faculties. And prior to this initiative, people would work in their own individual little worlds, and the level of collaboration that was necessary to make the true big impact on healthcare was harder to do. So now with the one university concept, I think we're moving ahead in a great direction. So I'm honored to work with the scholars who do so much tremendous work, many of them sitting in the room at the moment. And thank you for coming to be with us today. We're pleased to have the opportunity of having this prestigious lecture on the University of Calgary. And I'll now pass it over to Savraj Grewal, who will present. And he's an associate professor in the Department of Biochemistry and Molecular Biology, and he will present you. So thank you.
Thanks, John. So it's an absolute pleasure to be able to introduce Janet Rossant as our Friesen Prize winner for 2016. I've been asked to keep this brief, and so I just want to highlight two things about Janet. The first is as Janet as a scientist. Janet is quite simply one of the best developmental biologists, not just in Canada, but also in the world. Over her career, which has spanned now over 3 decades, she's continually made really fundamental discoveries about developmental biology, focusing on what is one of the most important questions, and that is, how does a single cell, a fertilized egg, give rise to all the various different cell types in an embryo? It's the question she's addressed in the mouse, and has introduced a number of really nice techniques to study that problem. And more recently, she's focused on a related problem, and that is how do stem cells give rise to their various cell types that they do. It's a body of work that has led to many really wonderful papers, all of which are kind of characterized by this elegance by which Janet and her lab has addressed these fundamental questions. And I urge all of you to take a chance to read these papers. It's also a body of work that quite rightly has been acknowledged by her peers, by the community. She is a Gairdner Award winner. She won the Conklin Medal from the Society of Developmental Biology, the Harrison Award from the International Society of Developmental Biology. She's a Fellow of the Royal Society of Canada, a Companion of the Royal Order of Canada. But more than all those things, it's a body of work that has really informed our current understanding of early development, of pluripotency, of basic stem cell biology. Things that are important for not just basic science, but also important for how we think about, how we understand, and how we can treat various human diseases. Which brings me on to my second point I want to make, and that's about Janet as a leader and an advocate for science. We think a lot about precision medicine, about genomics, our ability to sequence genomes in patients and start to characterize genetic mutations associated with disease. And at least in my mind, what we're starting to learn more and more is that what underlies these various genetic mutations are processes that are normally important for normal development. And it's the body of work and the investment that the scientific community has made into fundamental research over the last few decades that's allowed us to think about what these various genetic mutations may be doing. And undoubtedly, it's going to be our continued investment in fundamental research that's going to be important. And Janet has been one of the real voices, clear leaders in Canada advocating for the importance of fundamental research in basic science in health research. Just as a few examples, as the leader of the Sick Kids Hospital, she led one of the institutes, which I think is one of the best examples of investing in basic science and merging basic and clinical science as a way to think about and treat human disease. As a past president of the International Stem Cell Society for Stem Cell Research and the Canadian Stem Cell Foundation, she has thought not just about stem cell biology, but about the ethics associated with that. And she's been a really important voice in speaking to both the government and the public about, in a clear, knowledgeable way, about why we need to study stem cells and the important potential that stem cells have for for treating human disease, and I think you might touch upon that in her talk. And then finally, as John mentioned, she is the current president and scientific director of the Gairdner Foundation, which gives her an important role in recognizing fantastic scientists around the world and giving them these awards. But it's an important role in terms of outreach and connecting to the public, connecting to policymakers about why science is important and why we need to fund science and the important role that science plays, not just in health research, but in society in general. So I think we are incredibly lucky to have Janet as a part of the Canadian scientific community, not just for her scientific discoveries, but also for her leadership. And I can't think of a more worthy recipient of the Friesen Prize. So it's great to have you here. And I'd like to ask you all to join me in welcoming Janet to Calgary and to the podium to give her talk.
Thank you. Well, that was, okay, hang on. Thank you. That was quite the introduction. Thank you very much. I'm very flattered and honored by that introduction, and it's hard to live up to. However, what I think you heard in the introduction actually sort of encompasses what my talk is going to try to do, which is to give you a sort of view of my career and how the research interests that I have had have also led on to a career in investigating stem cells and how they might be applied to understanding and treating disease, and also into some of the public policy issues that come about when we think about human embryo work and human stem cells and gene editing. So I've called it From Embryos to Stem Cells to Gene Editing, a Personal Journey. And I'm going to start at the beginning. And the beginning, in this case, is my favorite stage of development. So we're going to start with some science. So those of you who are developmental biologists, this is for you. The next part, we'll go on to stem cells. So those of you who are interested in modeling human disease, this is for you. And then we'll end up with the ethics of gene editing. And those of you who are interested in public policy around and public ethical issues of stem cell research and human embryo research, that's for you. So hold on, there should be a bit for everyone. So, but for me, this is where it begins. This is what I have always been interested in from my early days as a graduate student, and it is 4 decades, not 3. This is the mouse blastocyst. And the mouse blastocyst has 3 distinct cell types in it. It's a small embryo. It's about 4 days to get to the stage in the mouse, 7 days in human, 100 cells, size of a speck of dust. And when we talk about the fertilized egg developing into an organism, the first thing a fertilized egg has to do is make the first cell decisions. And in a mammalian embryo, the first decisions are to make the trophectoderm, which goes on to make the placenta, as you'll see in a minute, the primitive endoderm on the surface here of the inner cell mass, and a small group of cells called the epiblast. And that structure, we now know, those 3 cell types are distinct lineages. But when I started working on this as a graduate student, we didn't have all these nice, pretty markers to mark these different cell types. We could look down the microscope and see that there seemed to be different morphologies. But we had a lot of questions. So we knew that we'd gone from an egg to a blastocyst. But what was happening in the blastocyst? What are these cell types? What do they give rise to? And when are they actually restricted? Are these really restricted lineages at this point? How are they established? How do we get from an egg to make those earliest cell decisions? And then, later on, as we started to understand that they are indeed distinct cell lineages, we asked whether we could derive lineage-specific stem cell lines so that from this tiny little embryo, where we don't have a lot of cells, we could develop cell lines that would allow us to have an endless supply of cells to study the lineage decisions that take place in the embryo. So, in fact, those questions that I started with as a graduate student, not the stem cell one, that was a later one, have really informed my research ongoing and have actually led my lab and many other labs around the world to realize that the mouse embryo turns out in our current research environment to be a wonderful system to not just study a process that is important for the development of a mammalian embryo and how human development occurs, but also as a system to get stem cells, but it's a wonderful basic biology system to understand at a single cell level how you go from an egg to cell fate decisions. It's a slow process. It takes 4 days. You can grow this embryo in a salt solution with a little bit of protein and an energy source. So it's self-organizing. All of these events are occurring within the embryo itself. No external signals. So it's a nice system to study self-organization. You can do single-cell molecular and cellular analysis and break down our understanding at the single-cell level. And increasingly, we can use in vivo imaging, live imaging, to watch the processes and watch molecules actually behaving in this process and understand how the lineages develop. And we're starting to see that we can use optogenetic manipulation to change cell fate. So I'm selling this to any of you who are interested in systems to understand cell fate decisions at the single cell level. And I believe the mouse embryo, above every other model organism, and I can have an argument with the people in the audience who work on other systems, the mouse embryo has some unique properties here. But going back to the original questions of what these cell types are in the blastocyst and what they give rise to, over many years, my lab and others has shown that indeed the outer cells go on to form the trophoblast of the placenta. Primitive endoderm makes the yolk sac layers, and it's this little group of epiblast cells that actually make the entire fetus, and therefore are called the pluripotent cells. So we showed many years ago that these epiblast cells essentially can make every cell type in the mouse and can make a whole mouse. So they are pluripotent, not totipotent, because cell fate is restricted. These epiblast cells, once you get to this late blastocyst, are restricted and can't make trophectoderm or primitive endoderm. So there's a 3-way restriction of cell fate. And over the years, we've had better and better methods to really dissect apart, again, at this sort of single cell level, this process of cell restriction. And over the last few years, a postdoc in my lab, Esther Postfai, has taken this question of when is cell fate restricted in the inner cell mass and the trophectoderm, in other words, when is pluripotency set aside from the cells that make the placenta, and really started to define in a temporal manner the windows at which that cell fate restriction occurs. And the experiments, I'm only going to summarize them very quickly here. You can look them up. It's just been published in eLife. But essentially what we've been able to do now is with the cell lineage traces that we have, we can isolate cells based on their likely prospective fate. And the marker we use is called CDX2. It's a gene, a transcription factor, that is expressed specifically in the trophectoderm and comes on as the embryo starts to undergo this lineage decision. We have a very nice GFP fusion to CDX2, so we can look at GFP and see exactly where CDX2 protein is occurring. And we can isolate single cells at different stages of development and sort them according to their level of GFP at a single cell level. And then what Esther did was to take CDX2 high or low cells and ask what happens if we put a cell that is already expressing GFP and is starting to turn on a trophectoderm marker, and put it back with an 8-cell embryo. Does that single cell contribute to the trophectoderm only? Does it contribute to the inner cell mass only? Or is it still capable of making both decisions? And then again, you can take the low cells, which we think would normally go on to make the inner cell mass, and ask how do they behave in this system. Now, it turns out that this assay is really reading out the normal cell fate of the cells in the embryo. Because what we see is that when we do that experiment, oops, so I'm running ahead of myself. Okay, let me just back up. So in this experiment, what we're seeing is that we're seeing a single cell, we're putting it with a morula, and what we find is that the cells behave in the same way they would in the intact embryo. And you'll see in a minute that that reads out the timing of specification of those cells in the embryo itself. But what we also wanted to do, what Esther wanted to do, was really test the full potential of these cells. Is it possible that a cell that has turned on CDX2 could be turned back into an inner cell mass progenitor? And ditto, can we take an inner cell mass progenitor and set it up in a situation where it's forced to turn on a trophectoderm marker? So the other set of experiments she did is shown here. In this case, she did the same thing. She isolated the single cells determined by their level of CDX2 expression. In this case, instead of taking a single cell, she took all the CDX2 high cells and put them together, or all the CDX2 low cells and put them together, and asked, in this situation, we're now forcing the cells to reveal their full potential. So all these CDX2 high cells, if they're in the inside, can they force back into inner cell mass? All these CDX2 low cells, can they reorganize and make a trophectoderm? So essentially, the cell has no choice. It can't re-find its position. It has to respond to position, and we read out the full potential of cells. And when we do that, we find an interesting fact that basically this assay reads out linear specification. It reads out what the cell would be doing in normal development. This assay clearly reads out on linear plasticity, when cells, although they may be going down a pathway, still retain plasticity. And what we see is kind of interesting, and this has not been seen as we've done other experiments in the past, that there's a difference in the timing between the inner cell mass and the trophectoderm as to when cell fate commitment occurs. And if we look at the trophectoderm, if we take a single GFP high cell and do this modular aggregation, if we take the cell from a 16-cell embryo or a late 16 or an early 32, and we put it in the combination here, those cells, despite the fact that in the intact embryo, they're starting to turn on CDX2, when we read out, they can actually still go both ways. And in fact, in the intact embryo, if we monitor the cell movements, they're still moving around. Things have not become finally fixed. However, by the late 32 cell stage, if we take a trophectoderm high, a CDX2 high cell, and do modular aggregation, it only ever contributes to the trophectoderm. And if we do the embryo reconstruction, the same is true. So in other words, even at this stage, although the single cell, the late 32 cell stage, is normally specified to trophectoderm, even if we take that cell and force it into the inside, it can't make the inner cell mass. So the cell is no longer plastic. So specification and commitment essentially coincide. When we look at the inner cell mass, it's a different story. What we see is rather similar to the trophectoderm. If we do this single cell assay, then again, by the late 32 cell stage, a single CDX2 low cell normally will only end up in the inner cell mass. So it finds its position, and this is the time at which normally the embryo starts to see less change between the outside and inside cells. However, if we do this embryo reconstruction, then at the early stages, it's the same. The cells can reconstruct an embryo with CDX2 low cells, makes both cell types. But actually, this continues longer. So even by the late 32 cell stage, when by specification, the cells only make inner cell mass, if we force them into this aggregate, then those outside CDX2 low cells can return on CDX2 and become trophectoderm. So there's a longer window of plasticity. The inner cell mass is not committed to fate until after the trophectoderm. And that's interesting because that leads us to ask, well, what is the mechanism by which cells are responding to their position in this gradual manner and ending up being restricted by this stage to one fate or another? And why is it that the inner cell mass cells are more open to this plasticity later in the trophectoderm? So what's the mechanism that drives lineage commitment? And this turns out to involve a signaling pathway, and it's the Hippo signaling pathway. And this is a long question that I and other people in the field had. Okay, we see this position-dependent specification. We see this timing of commitment. We know at the cellular level that these two lineages are being set aside. But what is it that makes that happen? And it took us a long while to get at it, I would say, largely because the Hippo signaling pathway that's critical here is a relative newcomer on the block. Not so new now, but in the last 10 years or so, we've known about FGF signaling, TGF-beta, BMP. Hippo signaling came from Drosophila, where it's a growth control pathway. Turns out to be important for growth control in mammalian cells. Many components of this pathway are tumor suppressor genes. And in this case, components of the Hippo signaling pathway have been reused, redirected to drive cell fate decisions in the early mouse embryo. And the clue to this came from an experiment that was done by a postdoc at the time, Henry Rossant, who's now an independent investigator at Michigan State, in collaboration with Hiro Suzuki's lab in Japan. They looked with we knew that there was that YAP, which is a co-activator of a transcription factor called TEAD4, was a key component in turning on trophectoderm genes. So TEAD4 YAP turns on CDX2. So it's a component that's required to make trophectoderm. But again, that just says, these are molecular markers. It doesn't tell you what turns on YAP, makes YAP and TEAD4 active in the trophectoderm lineage. The clue came from staining for a protein of YAP. Whoops, sorry, wrong one. And what we see, whoops, sorry, I had to go back one. What we see there is that YAP, the protein, is everywhere in the early embryo. And then as the embryo starts to make inside and outside cells, YAP becomes nuclear in the outside cells. And if you look carefully, it's excluded from the nucleus in the inside cells. So this is the co-activator of TEAD4. TEAD4 is required to activate trophectoderm. So if YAP is excluded from the nucleus, then we can imagine that this will ensure that trophectoderm genes are turned off in the inside and upregulated in the outside. And in fact, if you overlay YAP expression with CDX2 expression, I'm sorry that's not better projected here, you can see that these two overlay almost directly. So YAP nuclear localization predicts TEAD4 expression and predicts CDX2 expression. Over the next little while, Hiro Suzuki's lab particularly went on to show that the reason for this is that YAP is phosphorylated by a serine-threonine kinase called LATS, which is only active in the inside cells, phosphorylates YAP, keeps it out of the nucleus. In the outside cells, LATS is inactive, YAP enters the nucleus, and CDX2 is expressed. Now, this LATS is one of the key components of this so-called Hippo signaling pathway. And cutting a long story short from work from my lab, from Hiro Suzuki's lab, Magda Zernicza-Goetz's lab, many other labs over the last little while, we now are beginning to put together an understanding of what is happening that makes an inside cell different from an outside cell. It does depend, finally, on this YAP localization and the activity of LATS2. But LATS2 is only active if it's combined with a protein called NF2 and angiomotin at cell adhesion complexes in the inside cells. And in the outside cells, although ECAD and NF2 are still there, LATS is not able to combine with them and phosphorylate YAP because it's localized to the apical domain of these outside polarized cells. And it really then is this polarization of the outside cells that occurs as the trophectoderm becomes specified that ensures that there is a distinction and a commitment between the trophectoderm and the inner cell mass. Because polarity then is a key component, you can imagine that once this gets set up, the trophectoderm cells become specified and are unable to be able to reconstitute the inner cell mass. Whereas these inside cells, if they get exposed to the outside, they can repolarize and become trophectoderm again, beginning to suggest then that this would explain why trophectoderm is committed earlier than the inner cell mass. So do we have any evidence that that's the case and that Hippo signaling really is the driver of this difference in cell commitment timing? So that's the last set of experiments that Esther did. So we know that this drives inner cell mass trophectoderm segregation, but does the timing of sensitivity to Hippo correlate with the time of cell commitment? So to address that, we knew that we'd knocked out various components of the Hippo signaling pathway and we could activate it all over the place, but do it throughout the embryo in a not a time-specific manner. So what Esther did was to develop a set of experiments in which she could do a sort of temporal windows of activation or inactivation of Hippo signaling. So going from 16 to 64, 32, late 32, starting at different stages, she could either inactivate Hippo signaling by essentially inducing a dominant negative LATS2. Now, if you inactivate Hippo, what do you expect? It's a sort of backwards pathway. If you inactivate Hippo signaling, you will essentially block the ability of cells to form the inner cell mass. Everybody will become trophectoderm. The reverse is if you use ROCK-I, what ROCK-I does is activate Hippo signaling, And in that situation, you'd expect every cell to become inner cell mass, if they're capable still of responding. So the question is, at what window can these cells still respond to Hippo signaling? And the answer is quite clear, that if you're looking at the CDH2 low cells, the inside cells, which I said have this prolonged period of plasticity, then that prolonged period of plasticity correlates with their time at which they can respond to the inactivation of Hippo signaling. And that's shown here. So what we see is this is the time at which the doxycycline, which induces the dominant negatives, turned on, 16 cell, 32 cell, 64 cell. This is just a marker to show that we did indeed activate the transgene that's in these mice. And here, and I hope you can see this, this is just the marker again. But what you're looking at is if we inactivate Hippo signaling and the cells can still respond, then the inside cells should turn on CDH2. And that's what we see if we inactivate Hippo at 16 cell. If we inactivate it at the 32 cell stage, it's still the case. So that's that window of plasticity. But if you go to 64 cell stage and then activate Hippo signaling, the inside cells are committed and they can't change fate. So it does appear to correlate with the time of Hippo sensitivity. You would predict that the trophectoderm then would have a shorter, earlier window of responsiveness to Hippo, and that's what we see. So now, if Esther treats the embryos at different stages with ROCK inhibitor, that is going to activate Hippo signaling, and if the cells respond, then the trophectoderm, the CDH2 positive cells, should be turned off and become inner cell mass. And what we see is if you treat at the 16 cell stage, that indeed what happens is you turn down CDH2, you don't make a blastocyst, you end up with something that kind of looks like an inner cell mass. However, if you do that at latest, and that's what's shown here, you get this reduction in CDH2 expression. But if you do it at later stages, early 32, late 32, the cells don't respond. The trophectoderm cells will not change their fate. So we're beginning then by doing this combination of genetic experiments, timing experiments, lineage tracing, to realize that we do have the ability to understand at a single cell level the timing of events, the pathways that are important, and we now know that then we can link morphogenetic processes to signaling pathways to drive lineage specification in the early mouse embryo. And this summarizes a lot of experiments that I'm not going to go through in detail, but here just to remind you again that this first event is driven by Hippo signaling, and it's driven by position and polarity. The next event of making primitive endoderm and epiblast, which really sets aside the pluripotent cells, is actually driven by a different process that involves FGF signaling and random activation of FGF signaling in single cells that drives primitive endoderm versus epiblast, and then the cells sort out. So we're understanding now and being able to begin to model these processes of lineage specification. So to me, having studied the blastocyst, the mouse blastocyst all these years, to be able to begin to get down to this single molecule level and be really able to link molecular pathways with morphogenesis and cell commitment is really exciting and, again, a great model. Now we have to do it and begin to put more players into it and begin to build out computational modeling to understand at the single cell level how do cells interpret position, level of gene expression, and develop cell fate. But what it leads to is the late blastocyst. It leads to clearly committed progenitors at this point. All of our experiments, including our most recent ones, demonstrate that cells are committed to fate by this point. And that then leads us on to the next question and the next phase of the work, which had always been a long interest in my lab, of, well, if we have committed lineages, can we capture that commitment and develop stem cell lines that we can use to study lineage commitment in the Petri dish? And of course, the answer is yes. And the most famous stem cell lines that you get from a blastocyst, a mouse blastocyst, are embryonic stem cells. And I spent some time talking about the embryo because I want to remind you that in the embryo, the epiblast cells are pluripotent. They give rise to a mouse. They are not totipotent. They are restricted progenitors. They don't give rise to placenta and yolk sac. And that's exactly what you see with embryonic stem cells. If you make a chimera, they contribute to the fetus, not to the yolk sac, and not to the placenta. And over several years now, we've been able, in my lab, to derive trophoblast stem cells, which can also be maintained forever in culture. They have that expression of trophoblast markers like CDX2. You put those back in chimeras, they make the placenta, but not the fetus. And Zen cells come from the primitive endoderm, they express endoderm-specific markers, put them back in a chimera, they make yolk sac. So we do have in the mouse this possibility of capturing all three cell states of the blastocyst and retaining the lineage commitment that we see in the embryo itself. So one of the questions that comes out from this, though, as people developed embryonic stem cells, and we were not the first to derive mouse embryonic stem cells. This was work by Gail Martin and by Martin Evans and Matt Kaufman. But one of the questions was, are they really the same as epiblast cells? We know that epiblast cells make a mouse. If these cells are truly replicating the lineage restriction and the potential of these cells in culture, can they make a whole mouse? So are ES cells truly pluripotent? And one of the probably the most important experiments my colleague Andras Naj and I ever did was this experiment in which we tried to force embryonic stem cells to make a mouse. And we did that by what's called the tetraploid complementation technique. We knew that if you make a tetraploid mouse embryo, which you can do by cell fusion at the 2-cell stage, you just double up the amount of DNA, we knew that those tetraploid embryos, for reasons that we don't fully understand, but accept it, that they give rise well to the placenta and the yolk sac, but they're not very good at making the fetus itself. I told you that embryonic stem cells make the fetus, but they don't make trophoblasts and yolk sac. So we figured if we put these together, a bit like those reconstituted embryos I showed you earlier, you're going to force cells to take up the only thing they can do. If it's going to survive, the embryonic stem cells can only survive if supported by the tetraploid cells. And what we found in the first experiments we did is that, in fact, we could make an entire fetus from embryonic stem cells, provided that we had tetraploid cells contributing to the placenta. As time went on and cell lines got better, we're now, and indeed around the world now, people are able to develop mouse embryonic stem cell lines that are stable enough genetically, epigenetically, and every other way in culture over many generations, and show that in that assay, you can make live mice. So embryonic stem cells can make mice. So these are little GFP-positive mice derived entirely from cells that have been grown in the Petri dish for many, many generations. Still to be my most amazing experiment. And it does demonstrate the true pluripotency of these embryonic stem cells, their behavior as they would in the embryo itself. And it's really that paper gets quoted a huge amount. It's highly cited. And it's really, I think, cited for this reason, because it really says, well, if mouse embryonic stem cells can make a whole mouse, then human embryonic stem cells, we don't want to make a whole human out of them, but it does suggest that they would be a stem cell source that would have the capacity to make every cell type in the human, and therefore have this huge capacity for regenerative medicine. So, of course, human embryonic stem cells were derived much later than they were in the mouse, and that was by Jamie Thompson in 1998, essentially in a similar manner from the human blastocyst. They grow in culture. They are not identical in their properties. That's a different story we could talk about. To mouse embryonic stem cells, but they are identical in the sense that, at least as measured by their in vitro differentiation and by teratoma assays, they do have the capacity to make every cell type in the body. Going on from that, the next most important event that occurred in stem cell research was to go from human embryonic stem cells, which derived from human blastocysts, bringing their own ethical concerns, and certainly bringing a limited source of cells, to the concept of induced pluripotent stem cells, developed by Shinya Yamanaka. Now we're only looking at just over 10 years ago, so things are moving closer and closer, where he showed, first in the mouse, but then in the human, that you could take adults' fibroblasts and turn back the clock, and turn them into cells that seem to behave in the mouse exactly like mouse embryonic stem cells. They can make whole mice. And in humans, behaving like human embryonic stem cells and differentiate into many, many different cell types in the Petri dish. And the important thing here is two things. One, this gets rid of the ethical concerns with working with human embryos, but more importantly, it opens up huge potential to make pluripotent stem cells from many, many different patient and human sources. So it's possible, essentially, for everybody to have their own stem cells. And really, human-induced pluripotent stem cells then allow patient-specific stem cells to be developed. And they make many different cell types in the Petri dish. Importantly, they can, and it has been proposed, to start using them for regenerative medicine. So as pluripotent stem cells start to move towards the clinic, they are a potential source, and iPS cells in the long run would provide a tissue match source of cells for cell therapy. But perhaps more importantly is that the ability to take patient-specific stem cells means that we can model human development and human disease in the Petri dish in ways that we had no other way of doing before. And so this is where my lab, when I moved to sick kids, started to move slightly in a new direction. We spent a lot of time working on the mouse. We spent a lot of time working on mouse stem cells. We spent a lot of time talking about how important it was to do stem cell research because we could understand human disease and treat human disease. So at some point you go, well, I suppose we better do something about that. So the area that we chose to look at was this issue of modeling childhood disease, particularly genetic diseases, rare genetic diseases of childhood. Can we apply stem cell research and developmental biology, the two interests in my lab, to understand and perhaps potentially treat childhood disease? And coming from sick kids, the disease that captured our attention was cystic fibrosis. And cystic fibrosis, you all know, I'm sure, is a rare disease, not so rare, but it is rare, caused by mutation in the cystic fibrosis CFTR gene. There's a lot of interest, a sort of home run, as it were, at sick kids. The gene was cloned by Lapchi Choi, along with Francis Collins, in 1989. 1989, we've known the gene. It's a chloride channel. It's expressed in all epithelia, particularly in the lung. And its dysfunction causes inability to process, to really clear the lung airways, and it causes other epithelial defects as well. So it's a serious disease. The life expectancy for patients with CF has increased dramatically over the last years, since 1989, but all of that progress until very recently has been by treating the symptoms, better treatment of symptoms, and in fact Canada has some of the best survival rates in the world, and of course the final treatment is a lung transplant. So there has been progress, but it's still a disease with very high morbidity and impact on patients' lives. Given that the gene was cloned so long ago, why have we not moved forward knowing to have treatments focused on that understanding? Two things. Gene therapy started and failed. We'll come back to gene therapy in a minute. But now, and only now, new drug treatments are coming to the clinic that really do address the gene function. So for example, the major mutation that causes cystic fibrosis, the most common mutation, is an in-frame deletion where the gene is not properly processed to the cell surface. There are now drugs available that help move it to the cell surface and reconstitute function. Those drug treatments are going to the clinic, and they're going to the clinic at sick kids. They don't treat every mutation, and they're not always very effective. In particular, some patients respond and some patients don't. They're also extremely expensive. So there is a real push in this disease, as in many other diseases today, as we work towards new and important targeted therapies, we need to be able to develop means of making sure that the right patient receives the right drug at the right time, for the benefit of the patient, but also for the benefit of the healthcare system, that we cannot afford to be treating the wrong patients with these drugs. So we've developed what's called the CF Canada Sick Kids Program for Individualized CF Therapy. This is a wonderful collaboration between the Sick Kids CF Center, particularly Felix Rathjen, who's the head of the clinic, and Christine Baer, who's the research head, Center for Applied Genomics, CCRM, which is making our stem cell lines, and the Spartan Biocenter, which is doing the drug screening. And the idea here at this point is really to move towards precision medicine for CF. And we're building a library of iPS cells from CF patients. We have working towards 100 different CF patients coming through the Sick Kids Clinic. We're also correcting those, so we have controls. And then we're working to develop multi-well, high-throughput format assays for multiple cell types affected by CF in those patients, so that we can assess CFTR function in the dish and the response of those cells to the drugs that the patients might be receiving in the clinic. We match it with genomic, proteomic, and importantly, clinical data. So we know the patients are in the clinic receiving the drugs. We can compare and contrast what we see in the cells with what's happening in the clinic. And the goal here, we are still at the beginning phases, the proof of principle phases, but the goal is really to move this towards an individualized, patient-specific assay that we can say to the clinician, give us your patient material and we'll tell you which patient to treat with which drug. And a simple patient-specific assay at point of treatment. It's a multi-lab effort. Many of us across Toronto working on different ways of taking iPS cells and primary cells and differentiating them into the epithelia that are affected by CF mutations. And you see the list of players here. And you'll see I put myself at the top. That's only because I'm going to talk about that. My lab has taken on the goal of trying to make lung epithelial cells, the tissue that is most severely affected in CF, from iPS cells, and developing assays that we can use to mimic and understand patient responses to CF drugs in the Petri dish. And this was work from an ongoing work from a research associate in my lab, Amy Wong, in collaboration with James Ellis's lab in the IPS work, Christine Baer from the CFTR function assay, and Felix Rathjen, who is our clinical collaborators. And we've published this, and we're continuing to work on improving our ability to make lung epithelial cells from ES cells and iPS cells. But here shows you what we can achieve. Here's iPS cells. We direct them toward lung endoderm, and then we direct them further towards these tight junction coupled CFTR expressing ciliated lung epithelium. And just to sort of summarize how we do this, it depends on developmental biology. The only way we can go from an early pluripotent cell to a functional cell, whatever cell type it is, to mimic whatever disease, so we have to understand how those cell types arise in the embryo. And so in this case, we know that we first of all have to make them into endoderm, gut endoderm, the cells that are going to make all the endoderm lineages, and that requires a particular set of growth factors. We can then add other growth factors that will make the foregut endoderm, where the lung is going to arise. Another set of growth factors are known to be important to specify lung progenitors out of foregut. And then after that, we start to have lung progenitors, and we have a more complex culture conditions that I would say we don't fully understand, where we take known culture conditions that encourage epithelial cell growth from primary lung cells and grow our differentiated lung progenitors in the Petri dish. And then finally, to get this tight junction coupled epithelium, we need to assemble the lung epithelium as it would be in the lung, and that means at an air-liquid interface. And that polarizes and allows the cells to become a junctionally coupled epithelium. So we're doing that, and we're doing it consistently, and we're consistently improving this technology. It's not perfect yet. We can get very nice patches of coupled epithelium. You can see some of those actually here. These are just CFTR expressing cells from our differentiated normal cell types. But we are now at the point where we can do this efficiently enough that we can generate the cells also from CF patients. And you'll see in this case, no CFTR function expression at the cell surface. If we treat these CF cells with one of the drugs that is known to repackage CFTR to the cell surface, then we start to see CFTR coming to the cell surface in these cells. So we have proof of principle that we can read out drug function in these cells. And importantly, we've also started to now take patient cells with the same mutation and show that different patients will indeed respond differently to the same drugs. So we'll see here that one patient with the same CF mutation will respond pretty poorly to this drug. This patient, when we treat the cells, responds well. Now we need to combine that and see, does this actually match the clinical response in the clinic? So we're very excited about this, not just for CF, but for many other diseases, that there are ways now to use this to really start producing individualized therapies. So that's fine. We're modeling human disease, and it's a really important part of what we want to do. But of course, we're making lung epithelium cells. So aren't we also interested in the regenerative medicine aspects? Can we not use these lung epithelial cells to perhaps replace the damaged cells in CF and indeed in perhaps other lung diseases as well? So can we actually move from modeling the disease to correcting the disease? And so I'd like to end by talking a little bit about another game changer. We've talked about stem cells, we've talked about iPS cells and modeling disease. We are moving into the next revolution that's going to impact on fundamental research, but also potentially on treating disease, and that is genome editing. So CRISPR-Cas9 editing, I'm sure you're all aware, is an amazing tool that allows you to take a system that can edit genes in bacteria to prevent viral infection, and it's so efficient in providing a directed nuclease cut in the genome that it works across all species, including mice, mammals, humans, plants, many different animals, and it's highly efficient and highly specific. This means that we can do, and we do, edit and correct CF mutations in our iPS cells. That gives us our control cells. We know the mutation, we can very efficiently correct it and have control cells. We could, in theory then, take patients' iPS cells, correct the gene mutation, make them into lung cells, and perhaps treat the patient. Alternatively, and this is, I think, perhaps more directly important for CF, is if gene editing is so efficient, we should be able to introduce the gene editing tools in an AAV vector, have them infect lung cells directly in CF patients, and fix the gene in situ. We know that you wouldn't have to do it in all cells. 10% or so of cells would be enough to help those patients do better as they go forward. So gene therapy for CF starts to come into play as well. Not just introducing the functional gene and worrying about where it integrates, but actually fixing the gene and fixing it forever. And as you know, we talked about the Gairdner Foundation. It was my privilege at the Gairdner Gala last year to be able to host what I call the CRISPR-Cas9 Quintet. 5 awards were given to the founders of the CRISPR-Cas9 work, Rodolphe Barango and Philippe Horvath, who really were the people who were the foundation people working on the bacterial immunity system and worked out how CRISPR-Cas9 works in bacterial cells. And then Feng Zhang, Emmanuel Charpentier, and Jennifer Doudna, who are the people who showed that it could also work in mammalian cells. And Feng, in particular, has been the sort of the person who's really pushed it forward as a democratic tool that many, many people are now using for mammalian cell editing. So CRISPR-Cas9. It's a great tool. We can use it for research purposes. I'm sure many of you are using it in your own research. It has great potential for somatic gene editing, and we're seeing companies, clinical trials moving rapidly, very rapidly in this area. It's so efficient, however, that it started to raise another question, and that's the last part I want to finish off with, and that is, okay, could we actually use CRISPR-Cas9 not just to correct genes in the lungs of CF patients, people who have disease, go into those patients and fix them after the fact. Why don't we just fix it in the egg and have done with it? So if it's so efficient, why don't we think about germline gene editing and using gene editing tools to really fix genes in the embryo itself? In the mouse, we can do this, and we can actually do it very efficiently. So this becomes something that has to be considered. In the past, when we've ever talked about germline gene editing in humans, the scientists like me would say, well, it's all very well, but we don't have the tools, so don't worry about it. It's just not going to be feasible. That is not the case anymore. We do have the tools. It could be feasible. So it has raised in the public eye, again, this whole concept of designer babies. This is something I took off the internet. I actually added this bit at the bottom, which I think is the potential justification for germline editing, that you could actually go in and ensure that an embryo ahead of time was cured of cystic fibrosis, because we've been talking about it, perhaps more importantly, Huntington's disease. But what people also talk about is, well, if we could do that, why don't we go on and make designer babies? Now, you'll note that most of these things we actually, perhaps luckily, don't know how to do. We don't know the genes that are involved, so this is a way off. Sprinter, muscle, there are things you could think about doing for that. You could think about doing things to improve lung function. We could go in and make sure that everybody has the right ApoE allele that doesn't predispose to Alzheimer's. So where do you draw the line? And this is an ongoing debate. There are many organizations and agencies and governments and groups looking at and considering the ethical boundaries in their own context of their own environment and their own particular social environment. I think there's a general consensus coming about that the use of gene editing to actually study the biology of the human embryo in the Petri dish is allowable. That is to say, if this is a tool that will help us understand human development compared with the mouse, using this to really understand whether the mouse is a good model for human embryos, to understand how to improve IVF programs, to understand how early pregnancy loss occurs, all of that makes sense. And in fact, 2 groups have been approved to do such research, Kathy Nyeken in the UK and Fredrik Lanner, who's actually an ex-postdoc of mine in the Karolinska Institute in Sweden. So not in any way taking that to transfer back into mums, but just to understand the function of genes as I showed you in the mouse embryo. But there have been a lot of discussions, and I've been involved with them. This is me sitting on the platform at an international summit on human gene editing that took place at the National Academies of Science at the end of 2015. And coming out of that was a pretty broad-ranging discussion with many different stakeholders and advocacy groups, patient groups, ethicists, lawyers, clinicians, and also religious groups. But the overall consensus, and I put the red here, it's not coming up red on your screen, it's red on mine, would be irresponsible, which I think is a bit of a strong word, to proceed with any clinical use of germline editing until we understand safety and there's a broad societal consensus. And they felt that we weren't ready to do that at all. It wasn't a moratorium, it was a sort of, don't think it's good right now. It didn't say don't do it. After that summit, the National Academy of Science in the US set up a working group, so that was December 2015, a working group, again, of broad stakeholders. An international working group was also associated with the Chinese Academy, the UK Academy, the Wellcome Trust, to look at this issue and try to come up with some overarching consensus about where we should stand today and what we might see going forward around human gene editing. And I was part of that working group as well. It delivered its report over a year later, so it took us a while and very careful analysis. Oh dear, I just see that the next slide has gone screwy. Oh well, I'll have to explain it. And it was released on February the 14th. And you can read it, it's online, it's an in-depth document. And essentially, what it came up with, this is just a very brief summary, I would say these are the major recommendations, and I've given them red, green, and amber lights. Green light, genome editing for basic research, including embryos. So everything you're doing in the lab is fine. Even human embryos, they suggest, because it has fundamental information we can gain, makes sense. Somatic gene editing to treat disease, absolutely. We felt that the regulatory environment for gene therapy is easily transferable to gene editing, and that the positives here are enormous. Germline editing got an amber. Germline editing to treat disease when there is no alternative, and I'll come to that in a minute. And what got a red light was anything that smacked of enhancement, all that designer baby stuff that I just talked about, was considered to be, at this point, absolutely not something that should be done, because there was no way to really define the limits. So somatic or germline editing for enhancement was considered to be unacceptable and should not be approved under any regulatory framework worldwide. Somatic, I think, is going to, if you ask me where we're going to see enhancement, it's going to be somatic, and it's going to be in the sports arena. So how we regulate that's going to be interesting. But if we stick with the germline issue, then when we said, maybe, what were we talking about? So that the discussion ended up a bit like this. So caution is needed, but caution doesn't mean prohibition. So it was not intended to prohibit, but the point was to make healthy babies and not designer babies, which I think is a good line, and that these trials could be permitted, but only after we understand the safety and the efficacy, so general sort of understanding of the research process, under strict oversight, and that was not defined because different countries will have different oversight. It should be, however, restricted to prevention of serious genetic conditions, and for which there is no reasonable alternative. In some cases, pre-implantation diagnosis is a reasonable alternative. It's not available everywhere, it's not entirely clear what a reasonable alternative means, but I think the most important one is that it really was restricted to prevention of serious genetic conditions. Will we see it happen? Will we see it happen in the US? I think the answer is no. The FDA has actually prohibited from a rider in one of its funding things from Congress. It's prohibited from even considering this. What about Canada? Just end with Canada and just tell you where we stand in Canada. So where we stand in Canada is that there is a federal criminal law, the Assisted Human Reproduction Act, 2004, which, among other things, and some of my talks, I have all the other things as well that we're prohibited from doing, but this is one of them. It says, no person shall knowingly alter the genome of a cell of a human being or an in vitro embryo such that the alteration is capable of being transmitted to descendants. In other words, no germline editing. Punishable by a fine or 10 years in prison or both. So please do not start this in your basement. Interestingly, this two other things so it would prohibit that amber would be red in Canada. It would also be red for use of this to actually study human embryo development. And I've asked Health Canada directly on that, and their legal interpretation is that the line that says such that the alteration is capable of being transmitted doesn't mean you have to put it back to a mum. Just doing it so it is capable of being transmitted would mean it's banned in Canada. So in Canada, we cannot use gene editing tools on human embryos. It also prohibits mitochondrial replacement therapy, because mitochondrial replacement therapy, which involves nuclear transfer for patients who have mitochondrial disease, taking out and putting the mother's nuclei, the pronuclei of the father and the mother, into a donor egg. That's also prohibited because mitochondria have genetic material, so you would also be altering and transmitting alterations into the descendants. So we are actually prohibited from some of the applications. Mitochondrial replacement is now approved in the UK. The first baby was born in Mexico, so that is actually currently prohibited in Canada. So we are in an interesting situation. People think that Canada is a very open regulatory environment. It is for many things, but in this area, we actually have one of the more restrictive environments in the world. Good or bad, I'm just stating at this point that's where we stand. So we have an interesting situation. I think I've tried to illustrate to you that embryo research has informed stem cell research, continues to inform stem cell research in a two-way manner. Gene editing is moving apace. Germ cells can be developed from stem cells. Chimeras can be produced. All of this is changing very rapidly. Everywhere worldwide, the ethical, legal, and public policy implications are lagging behind, and I would say especially in Canada. So there is a need here and around the world to really think about how to engage the public, the patients, stakeholders, to think about where we want to draw the boundaries, and in Canada to think about how we deal with the legal environment in which we don't have an easy way of having a responsive system that changes to the changing science. So it's been a great career. This continues to be a great career. I haven't stopped yet. But it does, for me, it all begins and ends with the blastocyst. We keep coming back. It's a system that we can study at many different levels, and it's a system that leads me into stem cells, and it also leads me into public policy and ethical debates. Thank you.
So we're running a little bit late, so I'm suggesting to keep the questions confined to the reception afterwards. But we would like to hear just a few brief words from Dr. Albie Angel, and then we'll continue outside and carry on with the discussion, okay? It's working.
Good. Thank you very much. It's a great pleasure to be here once again, accompanying a Friesen Prize laureate. This is a very special event in Canada. As you can see, Janet Rossant has been an exceptionally productive scientist in evolutionary biology, in stem cell sciences, in translational medicine and advocacy, and now in policy development. She embodies all the things that the Friesen Prize program wishes to embrace, which is to support science, which is to communicate science to society, which is to support young people in their personal development, and also to advocate on behalf of the scientific community to our leaders to support research. You've heard much of her career. She's been introduced beautifully, and now you've witnessed a demonstration of supreme excellence in Canadian science. And I want you to join me in thanking Dr. Rossant for, number one, completing her responsibilities as a Friesen laureate, but also communicating so generously and meeting so many wonderful people here who's hosted. And finally, to thank the University of Calgary, particularly Dean Meddings, for being such a sterling sponsor of the Friesen Prize over the years. Thank you very much.